Okay, let's dive into this. Have you ever actually looked at a brain scan and wondered how in the world neurosurgeons and radiologists make sense of it? I mean, to the untrained eye, it just looks like a uniform gray maze of tissue. But how do they know exactly where critical functions, things like movement, touch, and speech, actually live? Well, it turns out it's not a maze at Story All. It's a highly organized map. Today, in this explainer, we're going to take a journey from the abstract anatomy of the human brain straight into reading real clinical scans. We'll be using a clean, evidence-based approach to decode these structures, taking what looks like a giant grayscale puzzle and turning it into a clear, navigable clinical landscape. Here's our roadmap for today's explainer. We'll start with the mapping question, then move to the cortical surface and Brodmann's areas. After that, imaging the medial surface, the mechanisms of the homunculus, the clinical takeaway of finding the hand knob, and finally, anatomical uncertainties. Section 1. The mapping question, finding functional landmarks. So before we can even start analyzing a scan, we have to understand the core problem we're trying to solve. How exactly do we map invisible function to physical structure in the human brain? The stakes here are absolutely crucial. If a doctor is planning a complex functional neurosurgery or just trying to establish baselines for a standard MRI, they literally cannot just guess. They need incredibly reliable ways to locate crucial areas. Take the motor strip, for example, which controls our movement. We have to find physical landmarks that perfectly correspond to these invisible functions. Moving to Section 2, cortical surface and Brodmann areas, the foundation. To answer our big mapping question, we have to start with the foundation. Think of the brain's outer surface like a beautifully detailed topographic map. All those complex folds you see, the sulci and gyri, they're divided into specific, shaded, functional territories. These are known as Brodmann's areas. This map basically breaks the brain down into distinct function zones based on cellular structure. It's kind of like the zip code system for the brain's outer surface, allowing clinicians to communicate precise locations without any ambiguity. Let's connect a few of these specific zip codes to what they actually do. Brodmann Area 4, for instance, is your primary motor cortex, the motor strip we just mentioned. And what's wild is that this area is packed with a huge concentration of these giant pyramidal cells called the cells of Betz. Just behind that, you've got areas 3, 1, and 2, which make up your primary somatosensory cortex. That's the area handling your sense of touch. Then, over on the dominant hemisphere, you have Area 44, Broca's area, classically known as the motor speech center. And all the way in the back is Area 17, your primary visual cortex. So you see, these aren't just random numbers. They are the fundamental coordinates of human experience. Section 3. Imaging the medial surface, seeing the data. Now, it's one thing to look at an abstract pastel drawing in a textbook, but we need to see how these landmarks actually show up on modern clinical imaging. If we want to localize that motor strip, we absolutely have to identify the central sulcus. And here is where the data gets really encouraging for clinicians. On axial imaging, the central sulcus is incredibly reliable. You can spot it on 93% of CT scans, but on an MRI, it's a perfect 100%. That 100% visibility means an MRI gives doctors an absolute rock-solid anchor point for navigating the geography of movement. But let's contrast that with another vital landmark, the pars marginalis. This is where the cingulate sulcus terminates at the back. On an axial scan, these paired grooves form this characteristic handlebar configuration straddling the midline. We call it a pars bracket. Interestingly, while they're visible on 91% of MRIs, they're actually slightly more visible on CT scans, coming in at 95%. So knowing exactly which imaging modality best highlights which structural groove, that's a real game changer for accurate medical mapping. Okay, so to make sure everyone is looking at these scans from the exact same angle, medicine requires a strictly standardized baseline. Enter the ACP line. This is the baseline connecting the anterior commissure, which is a white matter tract crossing in front of the fornix, straight back to the posterior commissure, another white matter band near the pineal gland. Setting this consistent horizontal baseline is absolutely critical. It's the standard used in functional neurosurgery, and it serves as the fundamental baseline for axial MRI scans and even recent CT scanners. Now, what's fascinating is seeing this baseline in action. Looking at the medial aspect of the right hemisphere, you can physically trace this dashed line, the AC-PC line cutting right across the middle of the brain. The specific line shown here uses what's called the Talairach definition. It anchors all the complex, squishy folds of the brain to a rigid, standardized, horizontal coordinate system. Section 4. Mechanisms. The homunculus. with our baseline set, how do specific human body parts actually map directly to these surfaces? Well, it brings us to the somatotopic organization of the brain. It's classically drawn as this caricature called the homunculus, which is Latin for little man. If you look at the primary motor cortex in pastel blue and the primary sensory cortex in pink, notice how the representation of the arm and the face are draped right over the outer convexity of the brain. Meanwhile, the foot and leg areas dive down, tucked along the upper part of the medial surface. Also, pay attention to the proportions. Areas that require intense fine motor or sensory function, like your fingers and your lips, have a massively larger area of representation. The map is scaled by complexity, not physical size. Section 5. Clinical takeaway. Finding the hand knob.
And this brilliantly illustrates how all of this anatomy and mechanism builds into one highly actionable clinical marker. Looking at an axial flare MRI, you can see exactly what clinicians are searching for. See that highlighted inverted blue omega shade projecting right into the central sulcus? That is the hand knob. The alpha motor neurons for hand function live right there in the superior aspect of the precentral gyrus. Because the human hand requires such an absurd amount of fine motor control, just like we saw with the homunculus, it literally bulges out into this distinctive knob-like protrusion. If you spot that omega shape, you have successfully mapped a critical piece of the patient's real-world function.
Section 6. Anatomical uncertainties, limitations in mapping.
Now, as clean and beautiful as these scans are, we have to admit, human biology isn't perfectly uniform. We have to acknowledge the clinical limitations. For instance, remember that absolute critical AC-PC baseline we established earlier? Yeah, there are actually competing standards for it. The widely used Taylor-Act definition passes through the superior edge of the AC and the inferior edge of the PC. But there's an alternative, the Schultenbrand definition, which passes straight through the midpoints of both. These two accepted definitions differ by almost 6 degrees. When you're trying to map millimeter-precise functional zones, a 6-degree tilt in your entire baseline is a massive variable.
And then you have pure structural anomalies. We rely so heavily on that central sulcus as our North Star, right? But anatomical texts show that the central sulcus actually joins the lateral sylvian fissure in only 2% of cases. That means 98% of the time it doesn't quite connect, and there's a bridging subcentral gyrus filling the gap. The map is an incredible guide, for sure, but the territory of every individual brain definitely has its own quirks. So what does this all mean? We've obviously come so far from the days of simply sketching the cortical surface, evolving to spot those inverted omegas on MRI scans with 100% reliability. But the brain is still vastly complex, full of individual variations and subtle, persistent structural uncertainties. So with all these anatomical variations and competing medical definitions still in play, how will the next generation of high-resolution imaging rewrite our maps of the mind? Thank you so much for joining me on this explainer, and keep questioning the evidence in front of you.